about each other. Yeah. Even if we didn't necessarily like, even if we weren't necessarily close friends. It's like, I know these people. Yeah.
Okay, how's everybody doing today? Ready to get started? Yeah? Monday morning, goodness. I was just talking a moment ago about the bad news that I have to share, which is that I missed the best half of my morning coffee, so you guys will probably have to suffer on the back end of that, but we're going to try to keep it interesting and fun. We have a lot to cover today. We're going to talk a lot about the neurobiology of memory. In particular, we talk about some of the introductory basics of synaptic plasticity. So hopefully some of you guys have already been familiarized with the idea of LTP, or long-term potentiation. We're going to revisit that to refresh our memories a little bit, kick off some of that dust and rust, and we'll hit the gas pedal and start moving a little quicker, start talking about some other neurobiological factors and how we could consider the synapse to be sort of this biochemical factory. Okay, and we'll talk a lot about what we mean, and again, just sort of how we finished off with last class, we'll pick up with the idea that we're going to introduce a lot of basic biochemistry and neurobiological terms, because our understanding of this stuff as we introduce it is going to set the groundwork and the framework for how we move forward in understanding some of the other biological aspects of memory, like the synapse. Make sense? Okay, cool. So we will cover that. Were there any questions on anything that we left off from, or the readings or anything like that beforehand? Everybody feels pretty good? Cool. Okay, so the other thing to note too, just ahead of time, we're going to cover basically that last page or two in chapter 2. We'll dive into all of chapter 3. On the syllabus, it jumps to 8, but next class we'll talk a little bit more about 4. So I just wanted to add that to the repertoire in case you're one of those good students, which I'm assuming everyone is, and wants to come to class prepared with a little bit of an idea of the content before I start yapping about it. Chapter 4, heads up. Hopefully help everybody pick it up, because it's going to talk about some biochemical terms. So how many of you guys have had cell and molecular biology before? Half a hand? Maybe a little bit?
Well, I had cellular neuroscience.
Okay, yeah, so that's pretty similar. How many of you guys have had cellular neuroscience? Okay, so hopefully this stuff will be some of the reviews. You guys know what like kinases are and phosphates? Okay, cool. So I'm actually aiming to shoot this at a basic level, but hopefully we might be able to amp it up. Hopefully this will be similar to a review then too. Okay, so we're going to be talking a lot about these ideas of synaptic plasticity, and we'll talk a little bit about introducing these. First, the idea of LTP as the sort of the neurobiological basis for learning and memory, and then some of the factors that affect LTP. So that's what we're going to talk about. These proteins, how they're arranged, how they're modified. We're going to talk about some of the other molecules that are involved in sending some of these signaling cascades involved with the proteins. And particularly, we're going to talk about how important different types of receptors for glutamate are in serving the basic neurobiological basis of memory, that is long-term potentiation. And then that'll give us basically a framework and a viewpoint from which to identify how each of these factors can be changed and affected in order to change and affect our memory, but also how they can go dysfunction, how they can how they can be impaired when we see dysfunctions of memory. Okay? So, without further ado, we'll talk about some of these facets, okay? So we finished off last class introducing this idea of LTP for anybody who needs to kick off a little bit of the rust and dust from our behavioral neuroscience classes. Remember, LTP stands for long-term potentiation. And last class, we finished introducing the idea of where some of our basic recording apparatus are placed for recording these things. Anybody remember which structure in the brain we identified for being a real key area for LTP? Where do we record this from? Yeah, the hippocampus. And what part of the brain is that in? Is it in the lateral temporal lobe? Yes. All right. So remember, you have the medial temporal lobe more towards the middle. The lateral temporal lobe isn't so much where the hippocampus is. You might get out to some other object recognition areas in that regard. But we're talking about the very medial area. So if we were to draw like a coronal slice or a brain, right? You have your ventricles, so on. You have your temporal lobes here, right? And our hippocampus, just roughly speaking, is going to be this medial area. Okay, and of course it's bilateral. And we'll talk about, when we talked about these different areas, you guys remember the trisynaptic circuit?
Mm-hmm.
Yeah? Information comes in from the perforant pathway, synapses on granule cells and dentate gyrus, project via the mossy fibers, CA3, project via shaper collaterals to CA1, and then there's output as well through the subiculum. Okay, it's a really good pathway to know. Now, we talked about some of those shaper collaterals as well, and how they're often a common site where we can begin to induce and measure and identify long-term potentiation. So basically, you stimulate the ever-living heck out of a presynaptic cell and makes a connection or a synapse onto our postsynaptic cell. Okay, these are generally both pyramidal cells from the CA3 to the CA1 area, and so we can record. Sometimes people record intracellularly, but we talked about this extracellular recording and why we can see an extracellular recording and infer postsynaptic potentials that are happening, right? You may remember the logic behind that? Yes, Francesca said if the ions are going in, then the outside is going to be lower. Let's expand upon that a little bit and talk about it in specificity. What ions, where, and why the heck does that matter? So what I'm looking for is basically a little reminding us of what are the charges of these ions, which ions are they, where in terms of intracellular, extracellular. So let's see if we can repackage that.
Positive ions going into the
Of
the CA1.
Yeah. And which positive ions?
Yeah.
Yeah. Shake it like a salt shaker.
Yeah.
Okay.
Yeah.
Yeah. Not a Polaroid. Salt shaker. Polaroid's a wholly different song. All right. Very, very different biological mechanism. So I try to think of fun ways that we can remember stuff. Sodium, salt. So shake it like a salt shaker. I forget who's saying that.
Yin Yang twins.
I'm sorry?
Yin Yang twins.
Oh, you did not forget that.
Yeah.
All right. Good fact. Okay. Maybe you can show us how to do that later.
No.
No?
No.
I had a student in class in behavioral neuroscience last semester. He attempted to gain extra credit by performing a dance of neural communication in front of our entire 100 and some odd person class. He was jamming out. He was actually blind and dropped. It was pretty good. Anyways, you're welcome.
Extra credit?
We did actually, yes. You're welcome to join the party.
Okay.
All right. Well, we'll keep the Yin Yang twins on the back burner for now. It's a standing offer. Okay. LTP. So we have our postsynaptic cells our postsynaptic potentials that can be measured here outside extracellularly of the cell, because our sodium ions extracellularly would be influxing on postsynaptic potential, right, which would give our positive. Typically, we might see that as a positive postsynaptic potential if we're measuring intracellularly. But of course, when we do that, we actually see this negative deflection when we measure extracellularly, and that's because of exactly what Francesca was alluding to. We have our this is our cell membrane, right? You have our phospholipids, and you have your sodium ions extracellularly. They're positively charged. They're influxing here, and so if you're starting off with all these positive charges, and that's sort of your baseline measurement, in comparison to our baseline measurement, you have that influx, which means these positive charges are going to be coming in, but yet you're measuring at this point extracellularly from your electrode. Of course, because it started off positive, you're going to see that as a relative negative dip. So we can see the slope come in. We talked about the idea of artifacts and that basic fiber volley when we first started making these initial recordings. And of course, artifact here, we're not talking about Indiana Jones and the Temple of Doom, or the Last Crusade, or whatever the first one was. But,
The Lost Star?
the what?
The Lost Star.
Raiders of the Lost Ark. How can I forget? Fortune and Glory. And so that's one of his lines from there. That might be too old for you guys. But the idea is that this is not an artifact in that archaeological way, but which is a good thing to find when you're an archaeologist. Here, artifacts are kind of like crappy stuff. You guys remember that technical term we used last class? CRAP, right? Commonly recorded artifactual potential. Okay. So when we do that, we can measure our postsynaptic potentials. And what we can do, and this is of course the underlying basis of LTP here, is we first create basically in log what we call this baseline response to a stimulus. So you basically measure that postsynaptic cell's response to a normal stimulus or whatever. And we can see them, we can track that here when we plot that, that basic EPSP, right, our excitatory postsynaptic potential. When we record that, we can measure this amplitude, basically how big and how much of a potential is there, how responsive is that cell. And normally we can say that we can plot its sort of baseline responsivity. Then you give it what's called an inducing stimulus. So you stimulate the ever-living heck out of it with a stimulating electrode. Okay, you zap it. Less technical term. Okay, and what happens is for some reason, this postsynaptic cell exhibits a differential responsivity than we know it normally offers. Okay? So, sample, 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 sample. You give a big induction of electrical stimulation, and then you continue to report how responsive that postsynaptic cell is. When you do that, we can see that these postsynaptic cells, postsynaptic potentials, exhibit a much higher responsivity, or greater field EPSPs. Okay? So what we see here is something changed. Okay? Remember we talked about the idea as a fundamental point of the class that memory is not something we can necessarily see, right? It's something we observe or infer, right? And we talked about the sort of framework for that being that learning and memory is thought to be evidenced by a change in behavior or a change in neural responsivity, right? So we just infer that if behavior changed somehow, that they must have learned or remembered something in one capacity or another, be it conscious or otherwise. Everybody good? All right, so we see this change in how the cell is responding. And what's interesting is that when we see this change, it persists for a pretty long time. And of course, it has to be higher than this baseline here. So this change from here to here is the change in the potentiation of that cell. But particularly important is that it lasts for a relatively long term. Of course, that gives us the long-term potentiation, or LTP. All right, so that's kind of our famous basic aspect of what's happening at LTP, and it's really useful because we're recording at basically the synaptic level. Okay, so we can make a very simple reduced system. Of course, it doesn't explain everything about memory, because memory, particularly in the types that at least I'm and I think that you're interested in, are the types that we experience, right, which require the interaction of networks of billions of neurons. So it's going to be far more complex than that which we can reduce to a simple synapse. But it gives us a good model to begin to ask and try to answer certain questions about what might be the biological basis that are underlying this. So when we look at this basic prep and this basic system, we can then begin to break it down systematically into what are the factors that might be involved in affecting this, observing this, okay? And so, again, we can sort of see this basic aspect of LTP, okay? And what we can also do is introduce this idea that we'll come back to and revisit, which is that LTP is not the whole story, right? Synaptic plasticity is bidirectional, okay? So, synaptic plasticity is bicurious. And what do we mean by that, right? It can go either way. It can be potentiated, or it can be really sad and depressed. All right, so we looked at it before, right? We had your sample, your test stimuli here, and then we gave our high stimulation here, and we saw that potentially. Remember? Well, we're looking at here something very, very different. Okay, so what happened instead of giving our high frequency stimulation. Now you get a low frequency stimulation. And that's kind of what this black bar is reflecting here. So if we came back here, that black bar might have been over here when we were stimulated. Okay? So that just shows when that stimulation is given. So here it's down here. And what we can see is that afterwards, not only is it not potentiated, all right, which meant that if it's not potentiated up here, the null hypothesis would be, well, if it didn't affect this, then the cell just shouldn't give a flying hoot. All right, it should just be right back up here. But instead, okay, what we see is that it's far below. Okay, so what that tells us is the cell is actually depressed in that capacity. And I'm not talking about a clinical psychiatric line or anything like that. Okay, but yes?
What's the difference between the black and the red?
Okay, so the black and the red, what we're looking at here in the legend, VEHAPV. Okay, so vehicle just means like your control, your normal type of thing. The way that people refer to this when you're doing these kind of studies, it's basically the application of a certain solution. So APV is particular, it blocks certain glutamate receptors. Okay, so you can put an APV out on NMDA, is how I try to remember it. So APV is basically a chemical, blocks glutamate receptors, particularly NMDA receptors, and we'll talk a lot about that in a couple more minutes. But the idea, very simply to your question, is that this is simply blocking some channels that people hypothesize might be involved in supporting memory. So when we look at these cells back here, it turns out that when they communicate, they're glutamatergic cells. So they use glutamate to communicate a lot, which means perhaps if there's something happening, some magic in the relationship, right, it's probably underlied by whatever it is that's serving the communication between those two cells. And we happen to know that it was glutamate. So then it gives us a target for certain receptors to look at. And we'll talk about there's 3 different receptors for glutamate, kainate, AMPA, and NMDA. In this case, they're blocking NMDA receptors with a chemical compound called APV. I can tell you the really long name. It'll only make you angry because you're going to think that you have to remember it. You don't. But it's a really long, crazy chemical name. I'm a cognitive neuroscientist, not a chemist. So, APV. So it blocks those receptors, and what it shows us is a couple things. So vehicle is basically our controlled normal situations, say application of water or saline or something basically benign or inert to the process. And that's where we see LTD, long-term depression happening, right? And again, the same idea. The cell gets depressed in its responsivity, and it extends for a long period of time. So you can imagine a time when it might have a very transient or short-term response where it might get depressed immediately, but then it returns back up here. That's not the case, though, is it? It extends long-term, and the same principle, right, for a long-term potentiation when it extends long-term, because it could, but then come back here. It doesn't. And so here we just see that when we block those NMDA receptors, that our long-term depression is no longer evident. So what that tells us is that somehow long-term depression is influenced or dependent upon NMDA receptor signaling of glutamate activity. Does that make sense? So this is where we start to move from just talking about LTP and sort of what it is, to start beginning to understand how it works by doing some of these experimental manipulations and controls. Make sense? Okay. Anything else on this? Oh yeah, so kind of an interesting analogy or example of LTD. So I was at the Society for Neuroscience meeting, gosh, probably 2 or 3 years ago, arguably more. And I was meeting up with a friend of mine from graduate school. And he was visiting from UC Davis. I was going up there from UT Dallas. And he was, before he went to graduate school, he had served in the Iraq War. He was a Force Recon Marine. So he was like a special operations, like really intense guy. And we used to go and work out and do long runs in Davis, because Davis is flat and that's just go run. There's not a lot else to do in Davis. You got to go outside the city a little bit. So we'd go and do runs and work out. And we were sitting there having a drink at this conference. And I was asking him, hey, how's it going? Or kind of BSing a little bit. And I asked him, hey, you still working out there, this and that? He's like, it's just been hard. He said, grad school's picking up. I got to work on the dissertation, this and that. Just normal chatting around about how work is going. And he said, it's weird. he's like, I haven't been able to really devote the energy to doing a lot of like intense workouts. He's like, and I can't seem to pick up the speed. I get stuck in this rut of like these long, slow jogs, and it ends up like, it seems like I can't actually make progress. And I looked at him, I'm like, yeah, you mean LTP? And he kind of thought about it, he's like, yeah, actually, yeah. So the idea is that, right, like when you have like high frequency energy or exercise, you really push it, you stretch yourself, right? We tend to grow. You stretch your muscles, they rebuild. This is the whole idea of weightlifting and weight training and all that. But then the idea, sort of this example from, if you stimulate very low frequency and all you do is just that low, you're never really going to stretch. And in fact, you might actually get a little slower and more tired. So just another way to think about long-term pressure. Okay, so we're going to talk about some of the factors that underlie this. So we're talking about this idea of the synapse as this biochemical factory. We're going to talk about the molecules that are involved, particularly in supporting it at a couple different levels. Okay, so let's erase this guy here. And okay, so we'll talk first a little bit about something called the extracellular matrix. Anybody know what that is? No? Yes?
Is that composition of proteins outside the membrane of the cell?
Yeah, so if we think about our synapse, right? You have your presynaptic cell here, you have your postsynaptic cell, or dendrite for that postsynaptic cell here. We have our dendritic spine. Okay, so this is our synapse here. And what we mean by the extracellular matrix are basically this conglomerate of all sorts of proteins and molecules that basically hang out in here. Okay, so I'm just put down a blue marker so I got another blue marker. Use Monday. Use black. All right, so it's the stuff that's in here. Okay, it's this matrix of things, and we'll talk about some of those factors and what they are exactly. So the synapse is not this empty domain as we've traditionally thought about it in, say, behavioral neuroscience introductions. But there's actually a lot of action happening there. It's very, very busy region. We'll talk about some of those molecules, and what we mean when we talk about this extracellular matrix, those molecules are coming from our glial cells. So hopefully you guys remember glial cells. Etymologically, they're coming from the Greek word for the glue that was supposed to just hold the brain together. They're supposed to be inert, sort of useless structural components. And of course, hopefully you guys all remember this new framework in which people have identified they're actually involved in signaling and a whole host of lots of other things. This is one of the things that they're involved in doing, producing some of these factors that are involved in our extracellular matrix. Okay, we're going to talk as well about something called the postsynaptic density. So you guys remember maybe, hopefully, some slides from last week when we introduced this, and I showed that there was this like darker shaded area. All right, so there's this postsynaptic density. If you look on cell slices or electron micrograph pictures, that just looks more dense. All right, now you can't really see proteins and molecules at that level of those pictures, but we were able to see that like shadowed area. You guys remember that slide? Mm-hmm. Okay, so we're going to talk about what that postsynaptic density is, and we're going to zoom in now. We're actually going to look at a lot of the proteins and molecules and chemicals that are involved and identifying what it is and also the role it plays critically in serving synaptic plasticity. That is the ability for synapses to change based upon experience. Okay, and so that postsynaptic density basically is comprised of hundreds of proteins, so it involves a lot of signaling enzymes, scaffolding proteins, molecules that adhere, basically are sort of the glue between other factors and compounds. And ion channels, those NMDA receptors we talked about, it's just lots of really good stuff is involved in that thick postsynaptic density, or PSD. So if you see it referred to as PSD, that's what we're talking about. Today is going to be kind of like an onslaught of abbreviations. It's like working for a government agency or something like that. Okay? So you're going to have like the FBI, the CIA, the DIA, the NSA, all those kind of good abbreviations, but now in our postsynaptic density in the synapse. Okay? Fun tidbit. So there's hundreds of these proteins, hundreds of types of them. A friend of mine got asked during her qualification exam for her PhD a couple years ago, which is like an interview or kind of like an interview, but it's really an oral exam for 2 to 3 hours. So it's 6 faculty who are like a firing squad that just ask you stuff, and all you have is like a piece of a marker or a piece of chalk, and you have to pretty much answer whatever they're firing at you. It's atrocious, right? But they asked us, like, how many proteins are there in the postsynaptic density, or in the synapse or whatever, and they were thinking about it in this context, just in the hundreds, but she was thinking more broadly. She just kind of looked at him, she's laughed, she's like, oh, thousands, there's thousands. And it was just frame reference. So, anyways, there's a lot of stuff in this postsynaptic density, and it serves 2 primary roles. One, it helps to basically localize our glutamate receptors and those adhesion molecules in the membrane. So what it does is it helps to hold our ion channels and other proteins and stuff. It helps kind of like provide that structural foundation, holding things in the membrane. And it also helps to position the molecules next to these. So we have, like, let's say a molecule. Helps to position them close enough so they can operate in a little bit of a speedier fashion when we want them to. Okay? So as you might imagine, there's a method to all this madness. They seem to have an organization. It's not just haphazard. So there seems to be good reasons for some of these things. Let's take a look at this onslaught of lots of biochemical stuff. Okay? Here's our synapse, because no one should have to suffer my artwork. Okay. It's kind of the same thing. It's a lot prettier. Okay? Just like we had over there, we have our synapse, we have our postsynaptic density, our ion channels, adhesion molecules, all that kind of good stuff. Okay, so here we have our extracellular matrix, which is shown by these basic gray kind of constructs. Okay, our postsynaptic density is this kind of area here, just like I drew in that horrible cartoon on this whiteboard. And then we're going to talk a lot about, particularly next class as well, about these structural components of the cell. We're going to call F-actin. Okay, and then of course we have our typical cell components. Okay, so we have smooth endoplasmic reticulum. We have our ribosomes helping to translate mRNA into proteins. Okay, you guys remember our central dogma? DNA, RNA, protein, transcription, translation, all that good stuff. Okay. We're going to introduce these ideas of what CAM kinase 2 is. Okay. And we'll talk a little bit as well about some of these presynaptic neurotransmitters within our synaptic vesicles. So we talked a lot about this stuff last class in terms of the presynaptic side of neurotransmission. So we're going to focus a lot of our stuff more or less on our postsynaptic side. Okay. And we'll talk a little bit about these kinases and the role that they play, and together with CAM kinase 2 and calcium influx and this F-actin, how they operate at the PSD to support these changes that we think are occurring in the synapse for learning and memory. Okay? So again, we can look in actual micrograph, we can see this thick postsynaptic density. See, some of my art's not really so bad, right? It looks the same way, right? They're both black. It's going to give me some points for that, right? Okay. Anyways, so that's kind of what we're looking at here. If we zoom in really close on this postsynaptic membrane, okay, we can see that phospholipid bilayer that I tried to draw about 5 minutes ago that was also atrocious. Remember I drew that little membrane? Okay, here we can see it a lot nicer. Okay, so we have our phospholipid membrane, extracellular, intracellular, and within this postsynaptic potential here, we can see that there's a whole bunch of stuff. Okay, so namely we can start sort of out on the outside. We have glutamate binding to our glutamatergic receptors. Okay, so we have glutamate receptors, and we already know that there's different types of glutamate receptors. There's ionotropic receptors and cladotropic receptors. This is kicking up a lot of dust and rust from earlier classwork, hopefully, the difference between ionotropic and metabotropic receptors. Okay, cool. What is a metabotropic receptor?
There's no channel.
There's no channel? Yes, ma'am.
It uses a G protein.
Okay, it uses a G protein, so it requires some sort of a metabolic process, right? Just metabotropic receptor. And so if an ion binds to it, does it open?
No, it sends the info to a G protein, which then travels to another area.
Exactly, so there's some sort of a second messenger system, as opposed to an ionotropic receptor, which operates how? Yeah, so I mean, they're both ultimately going to open a channel, but what's really the, that's how you would distinguish it, right? At a very basic level, right? We have the ion binds, channel opens, ionotropic. So that's kind of what we're looking at here, right? An ion, okay, it binds, and you have, it opens, whereas we can see a metabotropic receptor, it basically something binds and has to have a handful of second messenger or G protein coupled responses to open. Okay, so we already know that in our membrane we have those factors. Now remember these ion channels, they are themselves proteins. Okay, maybe we can draw that over here. So we can look at these ion channels in a couple different ways. One way is with our basic cartoon of an ion channel looking like that, right? Makes it really simplified, easy to break down. Very similar to what we have here. We have these 3 pink blobs. Okay, that's an ion channel, right? It opens up, whoop, ions go through. But of course, they're not just pink blobs, they're actually proteins. And the way that you can think about it is, instead, we can look at sort of, if this is our cell membrane, this is out, this is in, okay? They might be a 6 transmembrane protein. So you might have it where it crosses the membrane 6 times. 1, 2, 3, 4, 5, 6, okay? There's probably have intracellular domains. And so, but the idea here is this is a protein, okay? And it crosses the membrane 6 times, okay? And when you're looking at it sort of sideways here, let's look at a bird's eye view sort of like down, like we were looking through the tunnel, through the channel, okay? So now, what we can see is it kind of looks 1, 2, 3, 4, 5, 6 subunits. And now you can see where that ion channel, or ion itself, sodium, is going to go through sort of there to the inside from the outer. So the reason I wanted to say this and show this is just that these are proteins. And so it's something that I found it helpful to remember as I started to learn this stuff. Because those proteins, of course, are translated from the RNA, transcribed in the DNA. So we can see this expression of certain genes manifest as proteins that are put across the membrane to actually form and create that channel. Okay? Just like usual, my art is terrible, and I will try to show you some nicer pictures as we move along in the class. Okay? But it's one way to start thinking about these things. Okay, and so then we move down into the postsynaptic density, and we'll see that there's a bunch of stuff here. Okay, these basic proteins, molecules, stuff with SYNGAP, PSD95, all these connecting type of things. Schenck, Homer, basically connecting molecules and proteins, and they're going to form this postsynaptic density. Okay, so as you mentioned, one of the primary roles of this is to localize our glutamate receptors. So what localize would mean is basically put them there and keep them there. Okay, so kind of anchors. And again, it doesn't just anchor the glutamate receptors, it anchors some of the signaling molecules that can be put together with them. Okay, we'll talk a little bit about more of that as we move along. So let's talk about some of the steps in some of the signaling cascades. Okay, so we'll sort of work through this framework that when you have a first messenger, like a neurotransmitter, it's going to send something, let's say ionotropically or metabotropically, through a second messenger system. Okay, things like molecules, like calcium, IP3, other things like that. That's going to basically continue this sort of stream of communication. Proteins that are going to activate simultaneously kinases, phosphatases, that are ultimately going to lead to changes that happen in the synapse. Okay? They can be structural and functional changes in different proteins that are involved in creating this postsynaptic density that holds or changes our glutamate receptors in place. Remember that APV question that was asked about LTP and how it really clued us into the fact that NMDA receptors for glutamate seem to be very critical in this fundamental aspect of learning and memory. So we're going to talk about how these proteins, NMDA receptors, because these are proteins, how they're ultimately affected by this chain of events. Okay, so we're going to have these neurotransmitters operating as our first messengers. Okay, most of you guys should be familiar with this concept, right? We have serotonin, glutamate, all that kind of good neurotransmitter stuff, GABA, like the inhibitory for chloride ions. And then we're going to talk about some of these second messenger systems, right, which are going to be involved in relaying these signals. All right, these are more less basic chemicals like our first neurotransmitters, first messengers, or other molecules. And we talked about that as calcium, cyclic AMP, talk about IP3, other things like that. And so what we can do is we can begin to characterize this. So we talked about how this first messenger system initiates this signaling cascade, okay, such as glutamate, okay, or it could be a hormone that's binding to the surface of this cell membrane, and it's going to initiate, kick off this cascade, as we're going to think of it as, okay. Of course, the next step is going to be inherently the second messenger system. The second messenger system, as we mentioned a little while ago, cyclic AMP, IP3, and now the thing to remember is that these second messengers are molecules. They're not necessarily proteins. Okay? And the number of second messenger systems, okay, or these molecules, can rapidly increase when a certain neurotransmitter hormone binds to that postsynaptic cell, okay, particularly on the membrane. So one of the big benefits of that is that it allows a system to amplify the effect of the first messenger. Okay, so the reason that that says here that these second messengers are not proteins, why, if they're not proteins, why does that mean that they can amplify the effect of the first messengers?
Because they can activate other.
Okay, so they can activate other proteins, sure. But why do you think that's advantageous for them not to be proteins?
Because then they would increase the first messenger response?
It can't, well, indeed, they can definitely amplify the first messenger response, but why? Why faster?
They're less complex.
Okay, they're less complex. So what I'm thinking of is that they don't have to be translated. Right? So if we're a track coach, we've got our stopwatch and we're timing how fast all this stuff happens, right? And you've got the little runner around the track and it's a protein. It's going to be a pretty slow run because it has to go through it has to go through that central dogma, right? It has to be expressed from a gene, translated. I mean, this stuff takes a little bit of time. You have a molecule that's already there, bam, amplified. Right? It's a whole lot easier to have a molecule or synthesize it than it is to transcribe this protein. And the protein, now the protein is transcribed. You guys remember cellular neuroscience? Well, so what? The protein is transcribed. So now you've got to transport it into the membrane. You got to do all this extra stuff. Stuff takes time. It needs to be fast. Okay? So it's really good that these second messenger systems are not proteins and that they're instead these molecules. Because they can be rapidly produced, which means that when we need to amplify a signal, it can be done fast. Make sense? Cool. All right, so let's take a look at some examples of what we mean by that. Okay, so here we have our presynaptic cell, we have our postsynaptic cell, and all the players at hand here are our first messenger system. We'll also talk about some of our second messenger systems. Okay, so you have basically a shot of electricity comes in, and electrical action potentials, they come in through our presynaptic cell, they release. Okay, we just open our postsynaptic channels, influx of positive charges. Okay, what happens next is we have our second messenger systems. They're going to be rapidly deployed. Okay, and again, they're going to amplify the effect of that. So now we have our second messenger systems. That was calcium rushing in. Okay, and they're going to be able to amplify that effect. Okay, See that again? We have our first messenger systems coming in. There they are. Oh, sorry, I'm pressing the buttons too fast. Let's try that again. Let's go back here. All right. Action potential. Yes. First messenger systems. Influx positive charge. Now we have our second messenger systems. Come in. They can amplify that effect. So that's kind of the idea there. Okay? That amplification of the signal that we initially got from our first messenger systems. It's helpful to call your friends for help if you have to make this signal louder, right? A voice alone crying out in the wilderness. It's just our own voice, but we want to call our friends to add to it, and it enhances our signal. So a couple factors that we'll start to talk about are these phosphatases and protein kinases. So for anybody who's not familiar with these things, just a short introduction to them. So when we talk about protein kinases, they are basically enzymes that are going to be involved in modifying some of these proteins. And basically all it means is that you're adding a phosphate group to them. Okay, so this isn't a chemistry class, it's not a biochemical class. So I'm not gonna, I'm not expecting anybody to be chemists or to be able to work out equations or know all these details. But the simple point is to know that if we're talking about a protein kinase, they're adding a phosphate group to them, and what that ends up doing is basically phosphorylating. Okay? So what we mean by that is that basically it's engaging in some enzymatic activity. So we're changing the protein's cellular location or its ability to associate or bind with other proteins. Okay? So I'll leave it at that really simple level, okay? Because that's all we need to do for the purposes of this class. But when we talk about protein kinase, okay, we're talking about basically how it has two domains. And those two domains can be what we'll call inhibitory or catalytic. So when we're talking about something being catalytic, it's sort of that idea that we've talked about a little bit before, the metabotropic receptors, which is that it's engaging in some sort of energetic process. Okay, so it's actually doing something such as carrying out phosphorylation. Okay, and then of course I mentioned that we'll talk about phosphates too. Okay, and they're basically proteins that do kind of do the opposite. So they remove the phosphates from a protein instead of adding them. So very simple. Kinases are adding them. Phosphates are reducing them or removing them. Okay, and so the reason that we'll talk about these is because they're really important for basic regulation of some cellular activity. Now, so the cellular activity is going to be what's heavily involved in the synaptic changes that are underlying our neurobiological basis of memory. Okay, so we can talk about these kinases and phosphatases in a couple different ways. So we've already introduced the idea in our cell signaling cascade from a first messenger to a second messenger. Okay, IP3, calcium, these types of things. They're going to start working on some proteins then. So those proteins are then going to engage in these processes with kinases and phosphatases, okay? And so they can be either an active or an inactive state. Okay, so what we mean by that, let's see here. Yeah, we can have either an active or an inactive state. Okay, so this is the level of simplicity that we need for this kind of stuff, for the non-biochemists. We have two boxes. That's the level of complexity. Okay, they can either be in an active state or an inactive state. And that's it. Nice and easy, right? Cool. So, we're talking about our kinases. Remember I said that there's a catalytic unit to it, that's sort of active in that energetic process? Okay, usually it's inhibited by itself. Okay? Something that I have problems with. I could do so much better at everything in life. I would just inhibit myself a little bit on some things. I'm going to keep my mouth shut. Right? We engage in self-regulation. Okay? So it kind of works in that same way too with these kinases. We have a unit, it has a unit that basically comes over and blocks it from being able to do anything. Okay? But every now and then it can be dissociated and we can remove that inhibitory factor, and now that kinase is in its active role, in which case it can catalyze certain things. So let's go back to what we were talking about a moment ago. Where were we here? We did that. Did we do this? Yes, we did this. Why did it? Okay. Here we are. All right, so we talked about it can be an active or an inactive state. We'll come back to that in a minute. Okay, and how that inhibitory unit maintains it in that inactive state. All right, so that it's ready to operate when we need it to. And so the idea is that when we have that second messenger system coming in, okay, by virtue of this series of connections, that inhibitory unit on the kinase can be dissociated and activate that kinase in order to basically operate on phosphorylation. Okay, so you're basically phosphorylating the little proteins by adding that phosphate group. Okay, so what that means and what happens is you basically have the ability to change the function of proteins. Okay, so let's remove this. If this is our basic membrane, okay, be it through cartoons or proteins, either way, we have our channels. We have lots of proteins hanging out. This is our postsynaptic density, right? But just in our postsynaptic cell, we have all these proteins. Okay, they're not always active. They're not always doing stuff, but they certainly can do stuff. Right? This is what a lot of this phosphorylation is basically operating as. There's a way to activate them and get them to do stuff. And so that's kind of the idea, is that turns them into the active state and changes the function of our proteins. Okay, so by adding these phosphate groups, okay, by adding, let's say, our phosphate group there, this protein, and just for conversational purposes, can now be activated to do something. The simplest point. We'll talk about what that something is or can be along the way, but at a conceptual point, that's kind of what's happening here. And then, of course, we can have our phosphatases that come in and remove that phosphate. Okay? Phosphatase. You guys remember acetylcholinesterase? What does that do? Breaks down acetylcholine, right? So it's that ACE. ASE added to it. It's breaking down acetylcholine. Same thing here. Here's a phosphatase. I know this is like a lot of vocabulary and like stuff, so I'm trying to tie together ways to compartmentalize it. And you have a phosphatase. It's just like acetylcholinesterase that breaks down acetylcholine. This is basically breaking down the phosphorylation that had been done by the kinase. You all good? Okay. I'll post these slides for you and all that kind of jazz too if you need it, but a lot of it is very closely parallel to what you can dig it up in the text too. So we have that idea, and we introduce this idea that we have these kinases, and that there are these active and inactive ways. They can be dissociated, deactivated, or non-activated based on the phosphorylation of these proteins. Okay, and of course, the way that they can be activated from their inactivated state is by these second messengers. That's what SN is standing for here. So you have second messengers, they can activate and inactivate stuff. And we can take a look at more detail of how that happens. So here's our little schematic again. We have our presynaptic glutamatergic cell. Okay, we're going to have this zap. Our electrical potential or action potential comes down, it releases those cells with second messengers that amplify our signal, and now you have that dissociation of our kinase there. Okay? So we have our inactive, it was in our inactive state there, just sort of how we looked at it here, right? Nice and easy little cartoon boxes. Yeah? Okay. And when that happens, we can see the chain of events that are happening. Let's take that off. Remember that these are happening by virtue of our second messenger. Good? Questions? All right. How much time do we have here? Okay. What do we got? About a half hour? 25 minutes? Yeah, that seems about right. All right. Let's keep checking this stuff out. So we talked about these basically serving opposite roles of activating or inactivating based on their addition or removal of phosphate groups. So here, I had this little P here. And just like I always promise you, better art than what I can give you on this, my own horrible manual drawing. We have a nice cartoon piece of art that basically says the same thing. Okay? But hopefully it helps tie this stuff together. And so, see our basic protein here, right? Kind of looks like, actually, you know, I'm gonna stop putting my art down. You know, it kind of looks the same. How hard is it to draw that? It's the same thing, right? It's a good protein. Yes. So, we have our basically phosphate groups or kinases that can come and add or remove that phosphate. Okay, so we have the phosphate that can be added or removed, right? If you're going to remove it, you might use an ACE, like acetylcholinesterase, that's removing acetylcholine from the neuromuscular junction, for example. Okay, of course, the kinase might add it. Okay, because it's kind. When we're kind, usually people think that we're giving something to someone or adding something, right? It doesn't have to be the case. You can be, as you say, you can be probing kind, but we can think of kind being giving something, so we're adding something with the kinase. Okay, and that's kind of what we're seeing here. Okay, over there, activating it. Oh, can't stay on forever. All good things must end. Okay, phosphatase operates in that way. So we have this basic understanding. Let's dial in a little bit more. Okay, now here's our phospholipid bilayer, extracellular cell, intracellular part of the cell. Remember I mentioned that we have these transmembrane proteins, and this is forming our channel. Okay, so remember I erased that part a moment ago where it looked like that tunnel, or at least I hoped I was trying to make it look like a tunnel. You can imagine that's what it like if this rotated a little bit. You were looking down. Okay. So here's our basic glutamate receptor. Remember we're talking about glutamate because we're talking about LTP as a synaptic basis for learning and memory. Okay. There's several different subunits. Okay. And there's different binding sites at them for different neurotransmitters. When these neurotransmitters bind, our ions can come through the channels. Now usually they're closed, so they prevent our ions from coming through. Okay. So these are ligand-gated channels. So they open based upon the addition of a certain ligand that it happens to be put into its happy place by. Okay? Opens it up, those come in. Alright? You have that influx of sodium and calcium. Now we have a couple different ways we can characterize these receptors, okay? There are different receptors that can be characterized, I should say. We're talking about glutamate here. We mentioned earlier in the conversation, we have kainate, AMPA receptors, and NMDA receptors. Okay? So our kainate receptors, we're not really going to talk a lot about right now. They don't seem to be very much involved in LTP, but there's two major players in LTP, okay? And they are the life of the party. AMPA receptors and NMDA receptors. Can't have a party without these guys, okay? They have to be on the invitation. They got to be on the invitation list for different aspects of LTP. We'll talk about that. So AMPA receptors, okay? They kind of can be conceptualized like this. You can have ligands combined, okay? It influxes sodium, okay? So it's an excitatory situation here. Let's take a look at our NMDA receptors instead, okay? They're actually also permeable to calcium, okay? So they can let calcium flow through as well as with sodium, but hopefully you guys all remember from your behavioral neuroscience class, what's the role of calcium with NMDA receptors? Yes?
Is it in the neuromuscular junction?
Yeah, it probably operates in the neuromuscular junction. What I'm thinking about here is something about how calcium can or cannot enter.
Oh, magnesium.
Ah, yeah, magnesium who, what?
On the magnesium formula.
Clogs it up.
Ah, there's a magnesium blocker, is what we call it, right? So it clogs it up, is what you're saying?
Yes.
Most people talk about it in the context of magnesium blocker, okay? So it looks like this is real nice and easy for calcium to flow through here, okay? But of course, like most things we're starting off with is oversimplified schematic and characterization. Let's talk a little bit about some of the factors that can be at play here, okay? So when we talk about ways that we work on these receptors, and there's different ligands that combine to them, we have our normal situation where things are just closed and hanging out, right? Standard resting membrane potential, in a negative way, there's driving forces, different directions, different ions, and those channels are blocked, okay? What you can have is our typical situation, you have a neurotransmitter come that binds, it opens in an ionotropic way. Okay, everybody's happy, blah about this. You can also have something that we'll call an agonist. So here, basically is an agonist because the transmitter opens the cell. Okay, so this is a receptor agonist because you have a drug that can come and mimic that receptor, okay, or mimic the ion for that receptor and get it to open in the same way, okay. So hopefully you guys remember some of those drug contexts from behavioral neuroscience that you guys probably covered. And we can look at it in a couple other ways that things can affect the ion channels of these cells, okay? So we can block them, okay? You can have an antagonist. So it's kind of like anybody from convert from like a literature major or something like that, take literature classes, or dare I say high school. I never took maybe I did take a literature class in college. I forget what they had. It was the Stone Ages back then. We were writing on like stone tablets and things like that. But, I actually remember literature classes from high school, right? You have an antagonist, a protagonist, which is like the good guy. Then you have like the antagonist, which is like the twirly mustache, Mr. Burns character type of guy from The Simpsons hatching their evil plan. Okay, so same type of thing here. We have an antagonist that's going to interfere with the ability to operate on these ion channels the way our typical agonist or neurotransmitter typically would. So typically it would come in, it would block it, it would prevent it from coming in, therefore the channel stays closed. And you can have these things operate in other ways in which so this is a competitive one because they compete for the same binding site. You can also think about it in the way of a non-competitive binding. Okay, so here you have a neurotransmitter that binds to this receptor. Okay, now it doesn't competitively block it out. But what it does is it induces some changes in the receptor protein such that when the transmitter does come to bind, doesn't matter. Protein doesn't care anymore. Okay? So, different ways we can think about these ion channels being affected. So, let's talk a little bit about ways that people have identified some of the channels at play by using some of these strategies of blocking things. So like our APV example, okay? So some of this work was done by Ridge a little while ago. And let's take a look at breaking down what we're seeing on the screen here, okay? So we have 4 different plots of LTP. Okay, so we spent a lot of time introducing LTP earlier because it's going to be our medium through which we understand these learning and memory changes in synaptic plasticity by virtue of different experimental manipulations. Okay, so remember what VEH is? It's our vehicle, sort of your control analysis, control condition, in which we're going to see LTP normally exhibited. So our black line is our LTP normally exhibited. Okay, and when you use APV, we can see that it can block long-term potentiation. Okay, we can see certain situations, though, in which it varies based upon where this gray bar is. You guys what the gray bar represents in these figures is the time when you have that stimulation. Okay, so we're going to vary that. Okay, and we're going to be able to see that different things are affecting LTP in different ways for different reasons. Okay, so we're going to talk specifically about this NMDA receptor because remember I said we can put it, we're kind of like super troopers. We can put an APV out on NMDA. Okay, don't call me radio. Okay, super trooper fans. That's how I try to remember it. Put an APV out on NMDA. Right, it's a bunch of long letters. Okay, but this way we know the answer to your question. What is APV? Because it could work on AMPA, it could work on kinase, it works on NMDA receptors, and it blocks it in an antagonistic way, just like we introduced a few moments ago. And so one of the things that we can see here is that it prevents the ability for LTP to be induced, but it does not affect LTP on being expressed. So here's what we mean, okay? If you apply it here, okay, early, before LTP happens, okay, and you block NMDA receptors, you do not get LTP, okay? You just don't. We'll talk about some of the reasons why. However, what if you don't apply it here, and you just allow everything normal, and then bam, you apply APV. They don't care. Doesn't matter. So what it tells us is that somehow the NMDA receptor, which is what's being affected here experimentally, is involved in LTP, but only at certain times. So timing is really important. Okay, that's kind of the critical insight that we're gaining here. Okay, so if you do it early in time, it's very important and you don't get LTP. But if you first get LTP, NMDA receptors are not important for maintaining. Okay? Because you can block them, but the cell still remembers. You guys, is this making sense how we're sort of like really sort of systematically sort of going step by step into these factors? Okay. I won't be boring you guys. Okay. So let's take a look at another factor. Let's talk about those AMPA receptors. Okay. So remember, AMPA receptors are our ionotropic receptors as compared to our metabotropic NMDA receptors that we set the APV out for. Okay. AMPA receptors aren't going to be as responsive to blockage by APV. Some other crazy chemical, I don't even know what CNQX stands for. All I know is that it affects AMPA receptors, and that's all you need to know. Okay. We're not even using the name of NMDA receptors. We're just calling them NMDAs. So the abbreviations are very useful. I don't even remember what the whole list. Anybody remember what the whole string of NMDA stands for? No, you guys get like bonus points. You can remember that right now. You do?
I guess I may know the two first letters of NMDA. I think it's N-methyl. Is the last letter acetate?
It might be.
That no. N-methyl diacetyl something.
It might, but sadly, so you've gotten the bonus points. We need to know the answer, not the half of the answer.
Oh.
But it's a valiant effort, and I respect that.
Yep.
Good try.
Thanks.
Cool. But the point is that, right? We don't really need to know that because people in the field, we used to call it NMDA. We'll call it CNQX. And yeah, the important part to know is that it's an antagonist for AMPA receptors, not NMDAs. Okay, so now we're looking for the NMDA effects here. Now we're looking at AMPA effects. What does it tell you? Yes, AMPA is important for LTP. It does tell us that, but it tells us more than that.
Does it, the fact that the NMDA uses calcium?
Well, we can't know that right now based upon this experimental manipulation about calcium per se. But what did I say was the important point that we learned about this?
Maintenance of the LTP.
Yeah, I think I probably said that. I said something else though too. Oh, that's timing is important. Yes. Timing's important at the end of class because you want me to shut up so you can get out of here, right? You're like, dude, we're 3 minutes past class, right? Which is really crazy because, I mean, I take classes too. By the way, I mean, we never stop taking classes. They always want you to take classes, even when you're a professor, you go off and take specialized classes at other places and this and that. And when there's like 2 minutes past time, you're like, oh my God, why can't this end, right? Timing's really important then. If you're in the middle of class, 2 minutes ain't gonna matter to you. The timing's really important when it's 11:15 for us, and it's like 11:16, you're like, dude, come on, you gotta go. Okay, so timing is really important here in terms of when we apply NMDA receptor blockages for its effect on NMDA. What do we learn about NMDA receptors being important, but along that same line, what do we learn from here? Let's tell you.
AMPA's important too.
Huh?
AMPA's important.
Well, yes, that's what he said. AMPA is important. But what does it tell us about the timing? I'm sorry?
It was always important.
AMPA is always important. At both times. Right? Yeah. So, remember this is showing when that blockage is happening. Okay? But in both times, you don't get LTP. Here, you do. Okay, so what it tells us is that AMPA is necessary to induce LTP, but also to maintain it. Okay? Because here, you can induce it, right? Because this is normal, just like your normal, bam, block that stuff. As soon as it's blocked, what happens? It's gone. No mas. Okay? So it's important in both the maintenance and the initiation of LTP, right? And so again, think of this like a scientist trying to solve a mystery, trying to solve a puzzle. Like, we don't know what LTP is or how it works yet. You are the explorer in the laboratory trying to figure out what the heck is this process and what are the factors that affect it. These are critical insights that lead you down the right road. Make sense? That's why we're talking about this stuff in this way. Okay? So, let's look at things in a little bit more detail about what might be happening. We'll bring it back to this idea of the magnesium blocker. So, of course, we have our glutamatergic receptors. We have that magnesium ion that's blocking and preventing our calcium efflux. Okay? The way that things are thought to basically operate is you have our presynaptic release of neurotransmitter. It binds to both of these cells, both of these cell receptors. Okay? We have an adatropic, or we have an NMDA receptor and the AMPA receptor. Ions flow through. Now, how do we dislodge this magnesium blocker that's in there? Anybody remember? AMPA? It's what? No, teamwork.
The polarization of the inside of the memory should.
Yeah, so you depolarize the inside of the membrane. By depolarize, we mean make it more positive because we have more positive charges coming in. It depolarizes, and somehow it dislodges it by virtue of electrostatic forces. It's actually not somehow. Electrostatic forces is the way that it dislodges it. Right? What's electrostatic forces? It's a Paula Abdul song. Huh?
I don't know the lyrics to the song.
Like charges repel. Okay. Yes, but actually what her song is opposites attract. 2 steps forward, 2 steps back. Opposites attract. Alright, you can bring your guys back if you want. Alright, you gonna shake it like a salt shaker for us? Okay? No, okay. So then we'll stick with Paul Abdul. Opposites attract and likes repel. And so this is a positively charged ion, because there's a positive charge here, goes whoop, repels that sucker out. Once this is out, our calcium can come in. Okay? And that's kind of the idea there. Okay? And we have this NMDA receptor that basically is requiring these 2 events. First, there has to be glutamate that binds to the cell. Okay? So we have glutamate released presynaptically. Okay? And second, the cell has to depolarize, just like we talked about, so that there can be that electrostatic repulsion that dislodges that ion and allows the calcium to influx. Okay? Now, this is always the perceptive student, which most of you guys are, and I know that you're thinking this inside your head, which is, well, they're both positive. How come they're operating in different ways? Well, there's different driving forces upon them. So you got to remember your different concentration gradients and electrostatic gradients. So there's 2 different driving forces, and they can be differential in their application to different ions. Okay? So that's one thing to think about. Remember that. So we can see that here. We have that stimulation, release, binding, influx. Come on, influx. There it is. Okay. So it comes in. Our AMPA receptors are depolarizing our synaptic region. Okay. So now there's electrical difference here, and what happens is it ends up leading to that dislodging of that magnesium blocker. Okay. And now we can have calcium come in. Okay, so what we can see here is our biological manifestation of the LTP that we saw graphed based on the different factors. And what we said was some of the take-home messages of those LTP graphs was timing. Right? So we can see the biological way of how things are operating in different timing on these different receptors. And why, if you block each of them at different times, they can affect LTP in different ways that we saw exhibited on those graphs. Make sense? Okay. So, a couple ideas that we can take forward here is there's a real critical role of these AMPA receptors. Okay? So they're really important in both inducing LTP, because we can't get this to happen unless enough comes in through here. Okay, but we're also talking about they're really important in stabilizing LTP and keeping it persistent. Okay? And it gives us these couple ideas that people have begun to operate with. A lot of this came proposed from Gary Lynch in the 80s. And so the idea is that when you have LTP, this big zap of electrical stimulus, okay, it can rapidly increase how many AMPA receptors there are. Okay, and there's 2 different things. You can have basically constitutive recycling of these AMPA receptors. So this is the dendritic spine here postsynaptically, and you have these AMPA receptors, okay? And the idea with constitutive recycling of these cells, or these receptors, is that they're always being inserted and removed from the membrane. What we mean by constitutively is just being cycled in and out, okay? So you have these little vesicles that can insert the membranes or take them out. So see how this is kind of like bulging in, okay? It's endocytosing in, and it'll butt off and become this. And then something like this can feed back in, and these membranes fuse back in, and that's how those membranes are inserted into the membrane. Okay? Anybody had my behavioral neuroscience class, you know a little bit of how that happened with calcium-based synaptotagmin, SNAP25, what we call that kiss and run mechanism. Remember, we talked about that a little bit last class for presynaptic side, that same kind of endocytosis or exocytosis there is working on these vesicles as well. Okay, now you can also have signaling-based insertion of these in the membrane. Now what we see here is this different discoloration, this white part of postsynaptic density. So it kind of brings it home back to where we were talking about, because we need, for this stuff to happen, we're going to need to take this sort of postsynaptic density, which is kind of the glue or the cement that's holding all these structures together. By structures, we mean the proteins that are forming these ion channels. They're kind of cemented in place for good reason, right? We kind of want them where they are. But when there's learning and memory, stuff has to change. And this is what we'll talk a lot about more next class, is where the sledgehammers are and what the sledgehammers are that come in and bust up this cementing, holding this in place that allows the changes to happen. If we want to happen, if we want to change, because we want to be able to learn and grow from being an infant, right? So we need that stuff to happen. So you can rapidly increase those AMPA receptors, which is fundamental to expression of what we see as our LTP measurement. Okay. Now, another thing is that these AMPA receptors, of course, we also mentioned that they were trapped in and out of the membranes. We talked about how that's regulated constitutively, and as well by synaptic activity that can change that insertion of these AMPA receptors into the membrane back and forth. Okay, and it's the synaptic activity that stops to deliver the new AMPA receptors to those dendritic spines. Okay, let's see here what we have here. So one thing to remember is that timing is really important. So these synapses are strengthened based on the temporal sequences. Okay, and these are different even though overlapping processes. Okay, so we talked a little bit about this a lot. Let's see. Are there questions right now? A couple more minutes left, and I want to make sure that we have everything down here. So we can also see that that how long something is potentiated changes. So here we see that a certain thing is not, or a certain type of LTP doesn't last as long as a different type. And some factors that are involved in changing that is basically what we call theta burst stimulation. So basically these 100 hertz trains, okay, you can vary the parameters of that, and that's just what the colors here are. Blue is 1 theta burst stimulation, 1, 4, 8 changes how long that memory change lasts for. Okay, that's kind of the idea here. Okay, and we have this idea that we started talking about, that we can have this high frequency stimulation or theta burst leading to our LTP. Okay, and how that forms or leads to synaptic activity based on these biochemical interactions. We talked about, and we will talk about how it changes post-translational modifications. So it changes and modifies proteins after they've been translated. Okay? So remember our central dogma. Okay, so it's a little bit longer lasting than our immediate second messenger systems that we've talked about. And we talked about how we can actually increase transcription and translation of genetic expression into some of the proteins that make our AMPA receptors that then enhance those AMPA receptors' ability to operate in our cell membrane, and that ultimately strengthens the synapse. Okay, and that allows us to see the LTP when we measure in this capacity, or this, which looks nicer, but it's the same thing. Okay? So, and that'll lead us to where we're going to talk about in the next couple classes about how these synapses go through what we're going to consider 4 main stages, right? So you've got to generate that synaptic plasticity. You've got to generate that change. We talked a little bit about that today. We'll talk more about how that change can be stabilized, which means busting up that postsynaptic density, that concrete, with some sledgehammers that breaks it apart and allows it to then be stabilized, have those new AMPA receptors stabilized with newer concrete, for lack of a better word. And then we'll talk about how those memories can be consolidated and, of course, maintained long-term over time. That finishes what I wanted to say about that. Sort of, I guess, kind of sounds like Forrest Gump there. That's all I have to say about that. But before you go, what I wanted to do is actually 2 things. But if you have to go, you got to go. But what I wanted to say first is I planted the idea, hopefully last class, that that I want you guys to be thinking about topics and articles that you might want to present. Okay? I want to make things more interactive. So we have 20 people in class, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16 for people who already know this apparently and are at home studying the newer stuff because they're already experts, it must be. And we've got roughly about that many days of class meetings left. And for one question for you, would you like to do some article presentations on your own independently together as teamwork? With like a pair. Teamwork? Work together as a pair, share the load. Okay, so the idea that I want to be able to do is have you guys present for, say, an article that you think is really cool on a topic that you really like, and do that for about 20 minutes, and have everybody else responsible for engaging with questions and discussions, present basically a short introduction to the topic, 1 second, and then explains with us some of the methods and procedures that they were to use to capture the data, to address the introduction that of the topic, and then show some data what they did, and what they found, and then why is what they found really cool, or why is there some problems with what they found. And keep it to that kind of level and talk about it. And what I want to ask you is, would you like for me to assign articles, or would you like to go find those which tickle your fancy?
The latter.
You'd like to tickle your own fancy? Yeah. Okay. Then we will do that. What I will do is I will put like a Google document out that, or I don't know, is that even what they're called? A Google document? Sorry, I'm a bit of a Luddite. And maybe we can have signups. Okay? Does that sound reasonable, fair? Okay, so that'll be that. We talked a lot about synaptic plasticity and those aspects of the synapse. And so I'll leave you guys there. Let's see here. And so with that, I will hopefully, I don't want to keep you. If you guys got to go, you got to go. But this will be another way to think about the synaptic cleft.
So you're going to give us a list of articles to choose?
I'm sorry? No, you can choose your own. Anyways, have fun, guys.
We're supposed to, so each one of us are supposed to do the 20-minute presentation, right? Yeah. Is it supposed to be 20 minutes or how many? 20, yeah, you said 20 minutes. Right. I just really wanted to know if that's, if I heard it right. Oh.
You know I like this one. So.
I know you do.
It's kind of played out if you take a couple of my classes.
You have a partner?
Oh, yeah.
That's right. If I can't find anything,